Hello, and welcome to our video on clinical neurodevelopment with a primary focus on epilepsy. Epilepsy is a neurological disorder characterized by excessive or hypersynchronous electrical activity, which results in recurrent seizures. Depending on where these electrical misfirings can be localized to and how they spread throughout the brain, epilepsy can be split into two types, focal or generalized. Either localized, originating in a specific area of the brain, which is simple, without loss of consciousness, or complex, with impaired cognitive consciousness, or globalized, involving both hemispheres of the brain, including types such as tonic-clonic, absence, myoclonic seizures. Epilepsy can arise from a variety of causes, but it is strongly linked to neurodevelopment. The processes of synaptic and brain plasticity play a crucial role in wiring neuronal networks and determining overall network connectivity dynamics. Malformations in these can disrupt synchrony and connections, leading to excessive neural activity, which is a hallmark of epilepsy. This condition has a significant genetic basis, as seen in disorders like Dravet syndrome and juvenile myoclonic epilepsy. However, epilepsy can also result from other physical factors affecting these networks, such as structural abnormalities, for example tumors, malformations, and cortical dysplasias, metabolic imbalances that influence neuron excitability, and inflammation due to encephalitis or meningitis. Seizures can also occur idiopathically, which is without a clear cause. Now, kicking off the process of neuronal development, we start with neurulation. Neurulation is the initial step in the formation of the central nervous system, starting in the third week of embryonic development. The process begins with the formation of the neural plate, a thickened layer of ectodermal cells on the dorsal side of the embryo. Induced by signals from the notochord and surrounding mesoderm, the neural plate undergoes shaping and folding to create the neural groove flanked by the neural folds. Around day 22, the neural folds fuse at the midline to form the neural tube, a cylindrical structure that will eventually give rise to the brain and spinal cord. Fusion begins in the cervical region and proceeds both rostrally, down towards the head, and caudally, towards the tail, closing the neural tube completely by the end of the fourth week. Failure in neural tube closure can result in neural tube defects such as spina bifida or anencephaly. Spina bifida occurs when the neural tube fails to close properly at the caudal end, leading to various degrees of spinal cord and vertebral malformation. This is often with the spinal cord leaking out and being outside of the body, held in a sac. Symptoms range with minor physical disabilities to severe neurological impairment. Anencephaly is the opposite, which is a severe condition where the neural tube fails to close at the cranial end, resulting in the absence of major portions of the brain, skull, and scalp, which is typically fatal. Now, in epilepsy, some cases the neural tube defects can be associated with brain malformations that predispose individuals to epilepsy. For instance, individuals with severe spina bifida may develop Chiari malformations, which is associated with epilepsy. A Chiari malformation is a structural defect in the cerebellum that occurs when part of the brain bulges into the spinal canal through the foramen magnum, the opening in the skull where the brain and spinal cord meet, which can put pressure on the brain and spinal cord and lead to some of these mild or severe symptoms associated with later on epilepsy. The next part of neurodevelopment is patterning of the neural tube. Once the neural tube is formed, it undergoes regional specification through a process known as patterning. This involves the expression of different signaling along the anterior-posterior and dorsal-ventral axes. Anterior-posterior patterning of the neural tube differentiates into distinct regions through the influence of gradients of signaling molecules such as retinoic acid, fibroblast growth factors, and Wnt's. The anterior part of the neural tube expands to form three primary brain vesicles: the proencephalon, or forebrain, which further divides into the telencephalon, the cerebral hemispheres, and diencephalon, which forms the thalamus and hypothalamus. The mesencephalon, or midbrain, remains undivided and forms the midbrain. The rhombencephalon forms the hindbrain, which divides into the metencephalon of the pons and the cerebellum, and the myencephalon, forming the medulla oblongata. Dorsal ventral patterning of the neural tube is along the dorsal ventral axis under the influence of sonic hedgehog gene from the notochord and floor plane, which is ventral, and bone morphogenic proteins, BMPs, from the roof plate, which is dorsal. Sonic hedgehog induces the formation of motor neurons in the ventral spinal cord, while BMPs promote the development of sensory neurons in the dorsal region. Conditions that are often associated with this part of the development are prosencephaly, a disorder caused by the incomplete separation of the prosencephalon, or forebrain, into distinct hemispheres. This is often caused by mutations in the sonic hedgehog pathway and the gradients along these axes being altered. HPE can range from mild to severe, with severe forms leading to major facial and brain abnormalities. Another congenital brain malformation is Dandy-Walker malformation, involving the cerebellum and fluid-filled spaces around it. It's linked to abnormalities again in the dorsal ventral patterning, which can result in hydrocephalus, motor deficits, and epilepsy. Malformations of cortical development, such as focal cortical dysplasias, are related to disruptions in the neural tube patterning, and FCD is a common cause of intractable epilepsy, especially in children. FCD is a congenital abnormality in brain development that causes abnormal organization of the brain's layers and neurons. This occurs when the neurons in a specific area of the brain that doesn't migrate properly in the womb. The disorganization can lead to higher risk of seizures and disrupt brain function. FCD is the most common type of cortical dysplasia and a leading cause of drug-resistant epilepsy, especially in children, with 3 types of FCD. FCD can be caused by genetic and acquired factors, including mutations in the mTOR pathway, genes, and repositories of the signaling pathway.